Hello, and welcome back to this second half of the lecture for scientific ethics on recombinant DNA. And so in the first half, we talked about genetic engineering. We're combining DNA molecules or DNA fragments from two sources into kind of a single molecule, and then using that molecule to kind of change the characteristics of the organism that's going to be expressing it. So what we're going to do in part two of this lecture is take a look at some microorganisms, take a look at kind of small organisms that we can go through and do genetic engineering. And so again, to take a look at this from a perspective historically, what we can see is that microorganisms have been used for a variety of purposes for years. And so we may think about bacteria as being bad, bacteria cause disease, but if we take a look at it, bacteria have been used for a number of years, probably thousands of years now at this point, to convert milk into cheeses and yogurts. And so if you think about it, we've got a nice cheese platter over here on the right-hand side of this slide. All those little flavors that we, think about in the different cheeses are not there just kind of randomly. They're there because we've had a bacteria go through and, kind of digest some of the sugars that are present within the milk, and in the process of doing that, produce some byproducts, and those byproducts then can produce the flavors associated with cheese and yogurt. And so if we take a look at it, again, we say when we think about bacteria, when we think of bacteria, disease, maybe the need for antibiotics, but there are, kind of the scientific kind of factor, if we think about it here, you actually have about 1,000 different species of bacteria that are going to be in and on your body, and they're going to be there normally. And they're there actually to help you be a healthy individual. You've got some within your digestive system. They're going to help you, break down foods and absorb vitamins and things like that. And they're also important because they're going to be competing with those pathogenic or the disease-causing bacteria. And so that normal kind of collection of bacteria that's found within your body is important and needs to be maintained. If you think about somebody that is on antibiotic, a very powerful antibiotic, they often have digestion problems and they have to go through and eat, probiotics or a special type of yogurt to kind of reestablish those good bacteria within their digestive system. So we use bacteria for thousands of years. We've also used yeast. Yeasts are a different type of microorganism with the same basic idea. Yeast are a type of fungus. And so if we think about that, fungus and generally think bad, athlete's foot, things like that, mold, all kinds of bad things. But basically a fungus in the yeast, when we take a look at it, is something that's going to convert sugars into alcohol and carbon dioxide. And so the two yeasts that are most prevalent for human uses, we've got baker's yeast and brewer's yeast, the same basic idea. But we basically, take some of these yeast, mix them with the flour, and your bread is going to rise. You mix it with, grapes or other types of sugars, and it ferments into alcohols. So again, normal products, either bacteria or yeast, that we've been using for thousands of years for our purposes to allow us then to create kind of unique flavors and unique food sources for us. Now, if we take a look at bacteria, what we're going to see is that bacteria are continually evolving and changing. And the reason for that is if we take a look at that normal bacteria, we got a bacterial cell over here, we get the bacterial chromosome. Bacteria are very good at picking up DNA fragments. And in picking up those DNA fragments, they can be brought into the cell and many times incorporated into one of those vectors or incorporated into the main bacterial chromosome itself. And in doing so, it can pick up those traits. And so if you've heard of MRSA, that's a methicillin-resistant Staphylococcus aureus. Another great scientific name, don't worry about it, but basically it's an antibiotic-resistant bacteria. And it's a resistant bacteria that means that it's very, very hard to kill if it gets established within the body, because the antibiotics that we would normally use don't kill it. And so, that's something that happens normally if we take a look at these bacteria, which are capable of picking up random pieces of DNA, but picking up antibiotic resistance from that DNA from other sources. Now, if we take a look at this, bioremediation is this kind of this natural process where we try to break down pollutants into harmless molecules. And so if you take a look at something that is going to be biodegradable, this concept is that, you know, bacteria are going to be present, or something's going to be present within the environment that is going to break down whatever that material is, so that it can basically, you know, in a sense be recycled, but basically broken down so that it doesn't form like a plastic that's going to be sitting there for thousands of years. Now, the idea is that if we want to make more things biodegradable, we could go through and genetically modify bacteria so that they are going to be faster and more effective at breaking down these things. So going through, using this genetic engineering, using these, you know, techniques of recombining sources of the DNA to, you know, make new characteristics of the bacteria, we can change their characteristics and potentially make them better at breaking down pollutants. Now, an example of this is that some deep sea bacteria use oil as a food source. And so, you know, it's strange when we think about it. But bacteria can basically be used to break down oil spills. And so if we take a look at it, we've got this little beaker, we've got oil present in here. We put in some of these deep sea bacteria, and within 7 days, they basically have digested away that oil, and in doing so, done bioremediation, made that oil, which is normally not biodegradable, biodegradable. It was able to break it down, get rid of the pollution, and so potentially it could be used as pollution control. So a good example where that would have been helpful is the Gulf of Mexico oil spill in 2010. Massive spill. We had another one in Alaska, probably 5 or 10 years before that. If we can go through and treat the oil in a way that it's not, you know, glomming up all over the shores and on seabirds and killing fish and all of these other organisms, we can, you know, control pollution a lot better. And so again, this is an example of a normal organism which is present that we could potentially harness to do something for us. Now, to take a look at what we can actually do to change the characteristics of these microorganisms, think about this concept where we're talking about insulin. Insulin, we said, is important because it's involved with controlling the amount of sugar within your body. And patients that have diabetes are dependent upon having insulin as a source to control it, because their body's not producing enough insulin. Now, if you remember way back to when we were talking about organ transplants and things like that, we talked about tissue rejection. Normally, the source of insulin from about 1920 on was pig pancreas, occasionally cow pancreas, but pig pancreas insulin and cow pancreas insulin are different from human insulin. They have slightly different amino acids. They work very similar to one another, but it's possible for some people to mount an immune response and basically attack their insulin. And so basically they would get sick if they take insulin from animal sources. What they really need is human insulin. And now we're not going to go around collecting human pancreases and grinding them up to get human insulin. And so what we were able to do in the 1970s is basically go through, find the gene for insulin, put it into a bacteria, and have that bacteria produce insulin for us. And so we've got bacterial cells that are producing human insulin. And therefore, we're producing a form of insulin, recombinant DNA form of human insulin, which is not going to cause an immune system response. It's not going to cause problems for patients because it's human insulin. And again, this is a case where we've gone through, genetically modified a bacteria, got that bacteria to express the insulin gene, the human insulin gene, and produce human insulin. Another example of that is recombinant blood clotting factor 8. It's used for treating hemophilia, which is a bleeding disorder. Previously, prior to the late 1980s, patients with hemophilia, with this bleeding disorder, would have to get blood donations. They'd have to concentrate blood from multiple donors to be able to get the clotting factors that they need. If you remember, in the 80s, there was a high risk of infectious diseases such as AIDS, HIV, and hepatitis B. And so patients with hemophilia were at very huge risk for getting some of these infectious diseases. The ability to clone the gene for this blood clotting factor, put it into bacterial cells and get those bacterial cells to produce human blood clotting factor means that we have this new source of that without the need to take it from the blood and the risks associated with this. So what we're looking at here on the right-hand side of the slide are these stainless steel containers. And these are basically big vats filled with the equivalent of soup with bacteria that are growing in that. The bacteria are feeding on that soup, producing lots and lots and lots of the bacteria. And as those bacteria are growing, they're also going to be producing the blood clotting factor. We can isolate that, purify it, and use that for treating patients. So if we're taking a look at this, bacteria as a research model are often used in the laboratory, but we're learning more and more about how we can modify the characteristics of bacteria for a variety of human needs. You know, we've talked about it in terms of pollution control. So if we can improve the bacteria for eating oil, potentially we could go through and in an oil spill, spread these bacteria across the region. And, you know, in a period of days or weeks or months, instead of having a huge pollution and huge environmental impact, those bacteria would be able to digest away the oil. We talked about drug production for insulin and the blood clotting factor. Again, going through and getting human factors, human molecules that we can then use in patients without the risk of infectious disease, without the risk of an immune system response, because it's human proteins, we can better treat human disease. Chemical manufacturing, again, we can modify some of these bacteria to produce chemicals that we need for a variety of sources. And there's actually a movement to using bacteria as a biofuel. So instead of using oil and petrocarbons and all of that, gasoline as a fuel source, we can have the bacteria producing products that we can then use as a fuel source. And so this is, again, just an overview of microorganisms, primarily bacteria and yeast, that we can either use naturally or modify in some way and enhance their function, change their characteristics to allow us then to be able to better use these as like a little factory. So the readings for this, 2 readings. The first is a role of recombinant DNA technology to improve life. So the link for that. And then finally, a little bit more about CRISPR. Again, don't worry about all the science behind it. Just get a flavor for what they're doing. But again, this is incredibly exciting because this allows us to go in and make very specific changes to a DNA molecule at a level which wasn't possible even 5 or 10 years ago. So that finishes up the lecture. Come back for the next kind of mini lecture. We'll talk about the assignment or the activity that goes along with this. As always, if you have any questions, feel free to email me. Thank you and have a great day.